today we're going to do the second activity of our genetics unit, and it's really cool because we're going to be isolating our own DNA. And isolating DNA is one step in the making of a genetically modified organism, or GMO. By the end of today's lesson, I want you to be able to list and describe the steps needed to make a GMO, and we're going to do the one of isolating DNA, describe where DNA is located in cells, and list and describe the purpose of each basic step of DNA isolation. Let's get started with some notes. Follow along in your packet, and then we're going to do a little demo followed by isolating our DNA. So yesterday we got an idea about what a GMO is by looking at the example of BT corn. So how would you go about making a GMO, genetically modified organism? Well, the first thing you have to do is you have to identify a trait that you want your genetically modified organism to have. So you're going to identify a trait in another organism. Then you need to find out which piece of DNA or gene is responsible for that trait in the other organism. Finally, what you'll have to do is take that gene out of the first organism and put it into the DNA of a second organism. Now, these steps are pretty confusing without actually seeing how it's done. So what I want to do is look at the blueprint of one example of how scientists tried to make a tomato that wouldn't freeze. Let's take a closer look. So this series of graphics is going to take us through how scientists took a gene from a fish called a flounder that lives in very cold temperatures and put that gene into a tomato to try to make it so tomatoes are less likely to freeze. So let's take a second to read this together. The acronym GMO means genetically modified organism, which refers to plants and animals that have been modified by scientists in a laboratory. It starts with an organism that has a desirable trait, which is then combined with an organism that needs the trait. Try your hand at a standard fish and tomato modification. Alright, so the desirable trait is we don't want it to freeze. That desirable trait is in flounders, and it's going to be put into tomatoes. So here's step one. We have our flounder. Now, flounders look really cool. They're a fish that's really flat, kind of skates along the bottom. And in each cell of a flounder is DNA. And in each one of those cells is the gene, the piece of DNA, responsible for this antifreeze property. So, step one is to isolate that gene from the flounder, and let's see how that's done. After cells are taken out of the flounder, then the DNA is going to be taken out of those cells, and then you have to take the individual gene out of the DNA. And that part of the process is a little bit more involved, and it's one that we'll get into over the next couple days. So, extract the DNA, find the trait you want your tomato to have. In this case, we want the frost resistance of a flounder. So we've identified the gene that we wanted. We've taken that gene out of the organism, the flounder, and now we need to get that gene into tomatoes. So that leads us to step 3, transferring that DNA into seeds. Now this process is a little bit more complicated than this graphic suggests, but just for right now, imagine it's as easy as taking your extracted DNA and kind of like pouring it on tomatoes to get it into the tomato seeds. We'll get into a little bit more how this actually happens during this unit. After the DNA is transferred into the seeds, of course, you'll have to plant those seeds. So, those seeds go into the soil, you bury them, and make sure that they're getting proper nutrition and sunlight, and then what you have is a genetically modified tomato. Now, scientists did make this tomato, and it was called an antifreeze tomato, and a lot of people kind of freaked out about it. So they wanted to see if they could do it, but the antifreeze tomato was never commercialized, so it was never released to where people would eat it. Now, one of the drawbacks of having genetically modified organisms is that people kind of freak out, and instead of thinking about this as a tomato that doesn't freeze, they think about the tomato as being part fish and part tomato. That's the end of this video, but I just want to take a second to review how you make a genetically modified organism. Remember, step 1 is to identify a desirable trait in another organism. In this case, the desirable trait was antifreeze, and the other organism was a flounder. Number 2, isolate the gene from the organism that codes for the trait. And number 3, put the gene from one organism into another. I hope this video was helpful.